Hi guys, it's Mrs. Gary. I hope you had a great break. Today we're going to just do a little bit of review and then we're going to talk about translation. So as you recall, protein synthesis is when we go from DNA to RNA to protein. And remember that proteins make everything about you. They make all of your traits and control what goes on inside your body. And so the first thing we learned about was replication. We know that DNA replication happens in interphase of the cell cycle. Well, the next thing that's going to happen in order for these proteins to be made is remember that DNA is too large to leave the nucleus. It is double-stranded and therefore it's just a really big molecule and it can't fit out of the nuclear pores. And so what we have to do is we have to transcribe or change this message from DNA into something smaller, and that is going to be our messenger RNA. So our messenger RNA, remember, is single-stranded and it is small enough to leave the nucleus. And so that was what we practiced right before break, was changing or transcribing DNA into messenger RNA. And so today what we're going to be learning about is how do we get that code from DNA to messenger RNA to finally make that protein. And that process that we're going to be talking about is called translation. So I'm going to go through a couple slides here until we get to the right one. And so here is translation. So basically what we're going to be doing is we're going to be decoding the message of from mRNA into finally making a protein. So remember that translation happens after transcription. So mRNA is able to leave the nucleus and is transported to the ribosome. So you can see with this picture here that this is our nucleus, and these little tiny holes here are where that mRNA is going to come out. It's going to come out into the cytoplasm and head towards the ribosome where proteins are made. So translation, the process starts with a ribosome, which is made of proteins and a type of RNA called rRNA. And that R in RNA is really easy to remember because it stands for ribosomal RNA. A ribosome is going to attach to the messenger RNA, which is this blue strand right here, and here this gray area is our ribosome. It's going to attach to the mRNA and begin to read all of those letters that got transcribed from the DNA. And those basically are instructions on how to build a specific protein. And if you remember when we talked about macromolecules, amino acids are what makes up each of these proteins. So each of these little orange circles that I'm pointing to here are amino acids, and all of them together making this big long string is going to be a protein. And that protein is going to determine some kind of trait or activity within us. So all proteins are made of a specific amino acid sequence. So every protein in our body is made up of a certain sequence of amino acids. So you can see here that there's a certain sequence of amino acids. In case you weren't aware of this, there are only 20 amino acids. And you'll see that on the chart that I'm going to show you in a minute. But it's the order, the length, and the 3D shape of these proteins that determine what its function is. So every single protein is going to have a different order and a different length and a different 3D shape from each other, and that again determines what its job is. And so if you can remember this, these amino acids are held together by what we call peptide bonds, because proteins are sometimes called peptides. And so these little bonds that are here between each of these little beads, it looks like, or each of these little amino acids, is a peptide bond. When we have 1,000 or more amino acids, that is what makes a protein. And so when I was talking about the 3D shape of proteins, you can kind of see that. Sometimes they're a little swirly like this, a little curly Q, and sometimes they are flat like this. So there's lots of different shapes, and again, it's those shapes that determine what its job is. So let's first talk about how do we decode mRNA. So mRNA is read in 3-letter groupings called codons, and these 3 letters are read by the ribosome. Each mRNA codon indicates which amino acid will be carried to the ribosome by the tRNA. If you remember, tRNA is what we refer to as different type of RNA. And what that does is it brings in the amino acid to the ribosome to start making those proteins. So what I'm showing you on the screen right now, let me move this little picture here, down here. So this is one type of an amino acid chart. There are two different types, and I'm going to show you both. So the way we read this amino acid chart is first we look at the first three letters of an mRNA. And again, we only look at mRNA. We never look up the tRNA, okay? So that's really important to remember. So if we were to look up the first three letters, okay, remember those three letters are called a codon. If we look up these first three letters, A, U, G, on this chart. So I'm going to show you how to read this. So we start in the middle. Okay, so our first letter is A. So we're going to look at, see where my pointer is, see where the big yellow A is right here. This is where we're going to start. So that means it's going to be in this quarter of the circle here. And then our second letter is U. And so then we're going to go over here to this orange U here. Okay, and then the third letter is going to be in this darker orange area, which is G. And here's our G. And you can see here that this is the name of the amino acid, and it's called methionine. And it is usually abbreviated MET, and we usually always abbreviate our amino acids. And if you're not sure what the abbreviations are, you can always look them up. But methionine is what we call the start codon. The start codon is the beginning of every single protein. There could be other amino acids in front of that start codon, but the ribosome doesn't start reading it till it sees that MET or methionine. Okay, let's try another one. So here we have ACA. So again, we're going to look at this A. That's the first letter in this yellow A here. Our second letter is C in the light orange color here. And then we're looking for A, which is right here, and that is threonine. Okay, and it happens to have the abbreviation right here in the green area and the actual name of the amino acid in the blue. I would only ask that, sorry about that. I would only ask that you guys please just put the abbreviation. It's way easier. And no, I do not expect you to memorize this chart. You will always have it available to you. Okay, let's try one more. We have UAG. So let's try look up UAG. So U is our first letter, so we're going to look at our U on the yellow part here. Our next letter is A, so here's A on the light orange. And then G is right here in our dark orange, and you can see here that the word stop is here, and it's abbreviated STP. So what the stop means is it's not an actual amino acid. What that signals to the ribosome is that that's where that protein is going to stop being read by the ribosome. So it always reads from the start, which is the methionine, till it reaches a stop codon. Okay, and there are 3 different stop codons, and you'll notice that when you look through these. Okay, so I'm going to show you another chart that might be a little bit easier, and you can use actually either one of these charts, but I'm going to show you this other one just to make sure. And so we are going to look at these 3 amino acids again, or sorry, these 3 codons again, but use a different chart. Okay, so here is another amino acid. Okay, sorry, this is another amino acid chart. Hopefully you can see it well, and you can find a lot of these online. So let me move my picture over here. So the first one we said was AUG. So in this first column on the left is the letter A. Okay, so it says first letter, and that's the first letter of our codon, our 3-letter code. So we know that this first letter is going to be in this whole row somewhere. Okay, and it's kind of cut off here at the top. Let me see if I can move it for you. Here we go. The second letter up here is where we would look up the second letter. So we had A, and our second letter was going to be U. So we have AU, okay, and we know that it's going to be in this column and in this row if you look at where my pointer is. And then over here on the right side is where our third letter is. So we have to make sure we go down to the right section here. Remember, this is our A, our first letter, so it's going to be one of these 4 here. And if we look up AUG, G is the third letter, I'm going to move my pointer all the way across, and here's our start or methionine. Okay, So this is you can use this chart, or you can use the one on the previous slide. It's completely up to you. Most of the time I tend to give this chart, so I would try to get good at using this chart, and try to practice looking up several of these, and we'll talk about that again in a minute. So let's see here. So what's going on here is that the tRNA, which is right here, it's going to carry an amino acid, which is this pink guy here. It's going to carry that amino acid from the cytoplasm to the ribosome. Now on the other side of this tRNA is what we refer to as an anticodon. If you remember, the mRNA, the 3-letter code on an mRNA is called a codon, while the tRNA is called an anticodon, okay? And that anticodon is going to match complementary to the codon on the mRNA. So if you look down here where it says AAA, that means that the tRNA using, and remember this is RNA and not DNA, we're going to match every A with a U. So this one, if this is AAA on the mRNA, then this is going to be UUU on the tRNA, okay? And then that mRNA codon is actually the code for lysine, which is this amino acid. If we look at this next one, it's GAU on the messenger RNA, but it's on the tRNA, it's going to be CUA. And that is going to be this amino acid right here. And so what this code does is it makes sure that we have all of the amino acids and they're in the right order. And you can see these lines here, these are your peptide bonds, and we call this a peptide chain. So what ends up happening is that this tRNA drops off its amino acid, and then it goes back into the cytoplasm to grab another of the same amino acid and waits for it to come and match up with the codon again, okay? So here is it's going kind of fast, but here is a picture of the steps of translation. So mRNA codons are read by the ribosome, and the tRNA brings in the matching amino acid. Amino acids are strung together like beads on a necklace to make peptide bonds, and I've shown you guys that before. Here is that necklace, that beads of the necklace right there, okay? So if you can take a second to fill in this, or write this down on a piece of paper, fill in the mRNA nucleotides, or bases, I should say, the mRNA bases that are here. So like this first one, if this is T, then this is going to be A, and then A would be U, and C would be G. Okay, and then what you're going to do is you're going to look up the mRNA on that codon or amino acid chart and write down the first 3 letters of those amino acids. And then what you're going to do is figure out, well, which part is transcription and which part is translation. So take a minute, pause the video, and then come back and look at the next part of the video, and I will show you the actual answers. So here are the answers, and I've color-coded it for you. So we have our AUG on our chart is MET, UCA is SER, UGG is TRP, UAU is TYR, and UAG is stop. And so which part is transcription? That's DNA turning into mRNA, and which part is translation? When we go from mRNA to amino acids. So that's about it for this video. Please, please, please come and talk to me during office hours if you have questions. This can get a little complicated, but you do need to know this for your mastery project. So here is a great picture of the processes of protein synthesis. And I've also included a couple of videos here for you. So please make sure you go in and watch the video on protein synthesis, and there's also a video on how to read the codon chart. Again, if you have any questions and need help immediately, just email me, and then I can come on a live chat with you whenever you need me to, okay? Hope this was helpful, and I hope you guys have a great day and get to practicing, okay? Talk to you later. Bye-bye.